is growth control. And growth control is one of the most important things we worry about when we think about bacteria, because we're thinking about how do we control uh, the growth that we just talked about, but how can that help us preserve our own health or the health of the environment? Okay, so a couple of terms that we'll need to talk about growth control. The first is sterile. And sterile means that there are no living organisms. Uh, this is a very strict definition that even if there's one microbe in there, you can't have a sterile environment. Only when you have no living organisms do you have sterile conditions. Aseptic technique. In aseptic technique, this is a method of handling sterile materials so that we don't introduce any microbes into it, or so that they'll remain free of microbes. Aseptic technique is the way you deal with bacteria, say, in a laboratory. Okay, a few more terms. An antimicrobial agent. So an antimicrobial agent is any chemical that will kill or inhibit the growth of microbes. Antimicrobial agent is a broader term, and is anything that will kill or inhibit the growth of those microbes. An antiseptic is an antimicrobial that we can apply to living tissues. So something like alcohol, which is very good at killing bacteria, we can still use on our skin. Okay, a chemotherapeutic agent, this is anything that you can use internally. The first things that come to mind are antibiotics, and then it's perfectly fine to take an antibiotic internally, and it will kill them. And finally, an antibiotic is a chemical produced by a microbe that kills or inhibits the growth of other microorganisms. That term antibiotic is, again, very specific in that this is a chemotherapeutic agent which we can take internally in almost every case, and that is going to either kill or inhibit the growth of other microorganisms. In terms of non-chemotherapeutic terms, we can decontaminate, which means to make the surface of an object safe to handle. So we've reduced the number of bacteria or microbes in general to a point where it's perfectly safe to handle. A disinfectant is any antimicrobial that can only be used on inanimate objects. One of the disinfectants that we think about a lot may be bleach. Okay, bleach wouldn't be something you'd want to use on your skin, but you can certainly use it on inanimate objects. Okay, we can divide the killing into a few different forms, either physical forms or chemical forms. And the physical forms include heat, radiation, and filtration. The first of our methods for killing is with heat. And heat in general is a very easy way to kill things, and we'll be able to use it in a number of different ways. But the easy answer is the higher the heat, the more you're going to be able to kill things with them. And in general, the reason that that heat works is that you can denature all the macromolecules. And the most important of those to think about are proteins. So the proteins within the cell, if you denature them, they're not functional anymore, and so then you get the killing of that organism. As you look at the graph here, and so here we're looking at the fraction of the survival, and so we're starting off at 100% survival, and then at 3 different temperatures we're looking at how long it takes to kill 10% of them. 10% is decimal reduction time. So to go from 100% of the cells to 10% of the cells would be a decimal reduction time of 1. And so you can see that at 50 degrees it takes about 40 minutes to kill 10% of the bacteria. Raise it up to 60 degrees, and now in just 12 minutes we have the same amount of killing. And to kill 10% of the cells at 70 degrees only takes about 4 or 5 minutes. Okay, so one of the things that you can take home from this is that the longer it kills better the higher the temperature. So the lower the temperature, the longer it takes to kill. And in this graph, it's only from 50 centigrade to 70 centigrade, but is a real reduction in the amount of time to kill those 10%. Another real important thing about this killing is that this is going to be organism dependent. And so not just the broad sense of, say, bacteria versus algae or something that broad, but actually different types of bacteria will kill at different times. So something like an E. coli will in general die a lot sooner than something like Staphylococcus. Okay, so it's the amount of killing you need is going to be temperature dependent. Okay, and here's a bit of example of what I was just saying about different things taking different amounts of killing. So here we have an example of a mesophyll, or in other words, something that grows in sort of a medium temperature. Again, something like E. coli would be something like that. Or in B, a thermophile, and so this is something that likes heat. And so if we're looking at the temperature centigrade versus the amount of killing, the decimal reduction time, okay, at 115 degrees, say, the killing is pretty profound in something that lives at human body temperature, okay, a mesophyll. That same killing temperature in a thermophile would only be reducing about maybe 90% of the thermophiles. So those things that are able to live at higher temperatures in general are going to take even more heat in order to kill them. One of the forms that certain bacteria can survive in are endospores. And endospores are very heat resistant, and the endospore is going to be incredibly resistant to heat. So because there's so much less water in an endospore, it's a lot harder to kill things that are in the endospore form. And that's part of the reason why we sometimes have to do very extraordinary things. If the bacteria we know is contaminating something, and it's a sporulator, we might have to heat for a prolonged period of time or add more of the drug. This is a diagram of an autoclave, and an autoclave is one of the standard ways of sterilizing materials. So you have a two-chambered system, and you can put everything in through the door. Okay. Then once you seal the door, you can put steam in the outside jacket, the one in the darker red, and that will heat the inside chamber and also put a little bit of the steam in there. You always have to be watching the pressure, but these big autoclaves are sort of the standard of sterilizing things. This is one of the autoclaves at Stockton. It doesn't look that impressive just from the outside, but you have a chamber in here where you can put materials that need to be sterilized. It will record the temperatures and let you know how much pressure is built up in the system. And that pressure is what's important, or one of the things that's really important, because that's how we get the temperature high enough to be able to sterilize. So from a variety of organisms, it's been determined that 121 degrees centigrade is sort of your standard of where you want to get things to be able to autoclave them. So we can draw that we can start the steam in, and then it's going to take a bit of time to get up to that 121 degrees centigrade. Okay, and the objects inside of it will lag behind a little bit, both on the front end and on the back end. And what we need to do is we need to have a sterilization time, which is going to be 20 minutes of sterilization. So the gold standard of autoclaving something is to to sterilize for 121 degrees centigrade for 15 minutes. There might be some materials that are going to take a little bit more time than that, but it should work just fine. We can also sterilize things with radiation. And these can include many types of radiation, including microwaves, ultraviolet light, X-rays, or gamma rays. And how these things are working is they're killing by either increasing the heat, in the case of microwaves, or damaging the macromolecules for something like X-rays or gamma rays or UV light that will actually damage the DNA inside the cell, and you damage it to such a point that the cell can't survive. Okay. Here is a commercial laboratory size sterile air hood where you can see that we have UV lights in the back of it, and then sterile air is actually pumped through a system like this, and so then you can work on the surface and the area remains completely sterile. Now here's an effect of UV light on a plate of bacteria. In this case, it's called Serratia marciensis. And you can see that on the left side, on the right side here, this has been covered up with a piece of paper so that the UV light doesn't get to show onto it. This side, on the left, this was exposed to the UV light, and there's so many fewer bacteria on this side than on this side because the light was able to get in and kill them. And that actually illustrates one of the problems with sterilizing by ultraviolet radiation, is that it's pretty easy to cast a shadow so that you won't get the killing. Okay. Now the kinds of things that we can kill or we can use radiation with are things like tissue grafts or certain drugs that are temperature sensitive, or things like plastic where it might melt the supplies if you were to actually use the heat or an autoclave to try to get these to sterilize them. The tissue graft might be a little bit obvious, but any of those tissues, if you were to heat them to try to get the sterilization, you would probably cooked that either tendon or something, and that obviously wouldn't be functional anymore once you cook the meat. Okay. Our last method of sterilization is filtration, and in a way this picture illustrates this pretty well, and that is that the bacteria are going to be trapped on one side of the filter material, and that's how we sterilize something, how we get rid of all the microbes in it, by actually trapping them on one side of the filter. Here's another picture of something like that. This filter has very small holes that prevent the bacteria from passing through it. Okay, so you have to pick a filter that has the appropriate size holes in it so that the liquid will pass through it, but the microbes will be trapped on the outside of the filter. Now, in a laboratory setting or a medical setting, the filtration systems are fairly easy, where you'll have a membrane filter, okay, that is set down on top of a platform. It's going to go into a vacuum flask, and then we can put a little funnel on top of that, and so non-sterile media would go into the top of the funnel, and then sterile materials would come out the bottom. Okay. Here's a big picture of that. That we're going to put our non-sterile media into that top funnel, and we're going to get our sterile media once we use a vacuum to pull the air out of the flask so that we can get that media to fall down. Again, the bacteria are going to be trapped on the filter paper up here, meaning this media will be sterile. We can use chemicals in addition to the physical methods in order to kill the bacteria, and we can classify the chemical agents as either being bacteriostatic, bactericidal, or bacteriolytic. Now, when you're bactericidal, that means that you kill the growth, you stop the growth, but don't kill the cells. Okay. If you're bactericidal, now you're stopping the growth and killing the cells. And in bacteriolytic, you're going to just stop the growth, and in this case we actually lyse the cells. So if we look at each of these forms and how many bacteria we have there, so if you're bacterial static, and now we have the red line being the total number of cells that you could see in the microscope versus the number of viable cells, those that are still alive. In bacterial static, when you add the chemotherapeutic agent at this arrow here, those two things remain the same. The total number of cells you can see and the cells that are viable are exactly the same. In the static case, we're just trying to keep the cells from dividing more. Now, in a bactericidal system, the total number of cells you'll see remains constant, but the number of cells that are alive decreases. And so in the cidal case, now there are cells that are dying, and so the total number of cells, which when you look at it under the microscope, you wouldn't be able to tell the difference between live cells and dead cells. So the total number of cells would be the same. And in bacteriolytic, now we get a decrease in both the number of viable cells and the number of cells that you can see under the microscope, because lytic, again, is going to imply that the bacteria are lysing, okay, they're popping open. So as they pop open more, you're going to see less and less cells. A very common term that's used in microbiology is the minimum inhibitory concentration, or the MIC. And in this experiment, what they've done is they've taken the amount of antibiotic and decreased it in order in each of these tubes, and they're generally decreasing by half. Okay, so the point where you get no cells to survive is the minimum inhibitory concentration, or the least amount of that compound that we get absolutely no growth. Okay, here you can see a little bit of growth in this tube, so that's not the minimum inhibitory concentration, but the point where we see no growth, so no cloudiness in the test tube, is minimum inhibitory concentration. And the MIC is something you'll very often see on the paperwork that comes with some of your pharmaceuticals, that they will test, and they'll just report it as the MIC, and that's the minimum amount of that compound that you're going to take to keep the cells from growing. One of the methods to see how much, or compare maybe the killing effectiveness of microbes is something called an agar diffusion, or there's a special term for if you do it with just antibiotics. But so you take a plate of nutrient agar, and we put 5 mLs of a culture of bacteria that are in something called top agar, which is a very thin agar, and so that makes a nice even layer over these cells. We then put little discs with the type of antimicrobial that we want on it, then incubate 24 to 48 hours, and you can see a clear zone whenever that compound is killed. And so if you look at these two panels on the right, you can see that something like mercuroclate will have a very large zone of inhibition, So the distance from the compound to the edge of where there's any growth, that's a zone of inhibition, and it's quite large in some cases. For this bacteria, it's not quite as large. Okay, so this compound, or this bacteria, is much more sensitive to this compound. For iodine, here we have a reasonable zone of inhibition, but on the bottom bacteria we have almost none. Okay, so different kinds of mouthwashes will have different effectiveness against different kinds of bacteria. Different compounds will have different effectiveness against different types of bacteria. I was saying before that there's a special case when we use that disk diffusion assay for antibiotics, and that's something called the Kirby-Bauer assay. So in the Kirby-Bauer assay, the bacteria are streaked over the agar plate, and then paper discs with antibiotics, in this case, are put over that, then incubated, and we generate a zone of inhibition for each of the antibiotics. And this is a very, very common technique done in hospital labs, because if you saw no zone of inhibition, you would know that the bacteria were resistant to that kind of antibiotic, and so you couldn't use that as a drug. And that's one of the reasons why the Kirby-Bauer is done fairly consistently when someone comes into a hospital with some sort of infection, because you have to see what type of antibiotics that specific one, the one that is specifically growing inside of you, is going to be sensitive to. Okay, we have some chemicals that are either antiseptic or disinfectants, or in some cases both. Antiseptics, again, those things that we can use on our skin, things like alcohol or detergent, hydrogen peroxide, are all very excellent antiseptics. Okay, and saying detergents, it is pointing out that soap is a very effective antiseptic, which is why I'm such a proponent of washing your hands a lot. Okay, disinfectants, where you're just trying to clean a inanimate object, alcohol again, chlorine gas, or ozone, so O3, those are all very effective disinfectants. They can kill off a lot of things, but you can't use those on human skin. An interesting point here about the detergent and alcohol, you might know that your hand sanitizers are predominantly alcohol, and that's good. Okay, I mean, it's a perfectly fine thing to have on your skin, but one of the problems is that the bacteria they might be dead, but they're still there on your skin. Whereas if you use soap and water, you're almost always washing away the dead cells. So one of the things that I sort of don't like about hand sanitizer is that we have so many bacteria that are just there. Now, in a pinch, the hand sanitizer is perfectly wonderful. Okay, but I would still suggest that you wash your hands after using the hand sanitizer, say before you are going to be able to eat something. Okay, so you might be able to disinfect your hands real quickly with the hand sanitizer, but before you go ahead and eat something, you might want to use some soap and water to really get your hands clean. Okay, And this is just a big table of a number of different antimicrobials, and you'll see that all of them, they all are either solubilizing the lipids or the cell walls, um, or they can oxidize them, or they can denature things. And again, we have a number of different things that we can use as antiseptics. But here's a very specific example of how the concentration of different amounts of ethanol will affect the killing of the specific bacteria Streptococcus pyogenes. Streptococcus pyogenes is the causative agent of strep throat. And so what you can see that as you increase up to 95%, you're getting more and more killing. Okay, so if you're looking at the plus marks as being the killing, that at 95%, in 10 seconds, you're going to get some killing of the Streptococcus. If you were to drop that down to only 50% of the ethanol, then in 10 seconds you wouldn't get the killing. Okay. One of the real interesting things about this is that 100% is almost not effective at killing at any concentration. So you might see, a bottle of alcohol for cleaning your skin, and you might say, well, why isn't that 100% alcohol? And it's because the 100% alcohol just won't work. Okay, you actually sort of keep the cells alive. You fix them, which is something that we would do on, say, a microscope slide, but it doesn't kill them very effectively. Now that 50%, if you could do the treatment with the ethanol for over 30 seconds, then you're going to be able to get a decent amount of killing. And for the 40%, it's just too low to be able to kill anything. Okay, so we can, in general, increasing the concentration over a certain range will kill more effectively. And here's just a graph of some of those killing patterns. And remember I told you that soap and water is a very good antimicrobial, but you can see that it might at best only be about 90%, and that's at 120 minutes. That's in general pretty good, and one of the things that you can do to sort of help yourself is to always wash your hands with soap and water for about 2 minutes, or in other words, the amount of time it takes to sing happy birthday twice. Okay, so as you're washing your hands, sing happy birthday to yourself, sing it twice, and then you're probably getting to that 90% point where you're getting the vast majority of the cells to die, certainly for making it safe enough to be able to say eat or something like that. And things like acetone or iodine are going to be able to disinfect a lot quicker with lower concentrations, but iodine isn't as safe on your skin as something like just soap and water. Okay. So soap and water is a very good disinfectant. You're getting the amount of bacteria off, and you're also the physical action of washing it under water is going to help you, but you have to do it for long enough to make it effective.